Welcome to the stage, Muriel Dewilde from Biobase Europe Pilot Plant.
Thanks. Thank you so much, Paul, and your team for organizing this nice stage and bringing this beautiful crowd together in Omaha as well as The Hague. Grateful for that. But for now, I'd like to take you to Ghent in Belgium, to the Biobase Europe pilot plant, to talk about an interesting and very promising group of molecules, proteins. So we've been at this scale-up for 15 years. We have a track record of serving companies to develop, scale up, and manufacture their innovative bio-based products, turning grams into tons, basically. We do that for the entire bioeconomy. We're a state-of-the-art pilot and demonstration facility to produce all kinds of molecules, the microbial proteins and fats, specialty carbohydrates, natural colorants, ag-tech products, chemicals, bioplastics, enzymes, flavors and fragrances, you name it. And for that, we have a very wide range of state-of-the-art equipment. The heart of our plant is about fermentation. So we run batch, fed batch, and continuous fermentation processes. We grow bacteria, yeast, fungal strains, microalgae, typically in aerobic conditions, but sometimes also in anaerobic fermentations, even gas fermentation. We can feed them with methanol, specifically if we want to produce proteins at all scales. And the scales I'm talking about are bench scale. We have 25 fermenters at the bench. And then we quickly grow into 30-liter scale, 150-liter scale for feasibility testing, optimization. We have a special Fast 500 DAB reactor that was mentioned earlier today. We have 5 1.5 cubic meter fermenters that we typically use to make regulatory batches. Then our 15 cubic meter fermenters, real workhorses that we've used multiple times in the past 15 years to produce hundreds of kilograms or tons of material, first of a series productions, and our famous fermentation park 75 cubic meter fermenter that you see here at the bottom. Of course, after the fermentation comes the downstream purification that needs to be matched at all the different scales, and we need to have all the unit operations in place to purify all those bioeconomy molecules. So we can do cell lysis, we have high-speed centrifugal separators. We do a lot of cross-flow filtration, micro, ultra, nanofiltration, ion exchange, activated carbon treatment, crystallization, chromatography, solvent extraction, evaporation, crystallization, and drying, just to mention that we have all this equipment at all scales in place. And we help companies grow from lab to fab. Sounds very fabulous, but actually a lot of de-risking, troubleshooting, and issues along the way that you need to overcome to be able to be successful at this industrial scale. Back to proteins. For which companies did we do this track from lab to fab? Enough, Scottish company, producing a fungal meat replacer. Arbion, a French company, producing a single-cell protein from woody sugars. Onego Bio, a Finnish company producing ovalbumin via trichoderma fermentation. Those Vegan Cowboys, a Belgian company producing casein. Vivici, beta-lactoglobulin, Dutch producer. Biotalis, a Belgian-Flemish company for whom we produce antibodies as fungicides in ag-tech applications. All proteins, but very diverse products and very diverse applications. We also started the Protein Club to strengthen the ecosystem of microbial protein producers. Our partners in crime are ILVO and the University of Ghent. And we also started with this Protein Club a project called Firm Protein, funded by Flemish Innovation and Entrepreneurship Fund. Within this project, we are building a value chain tool. That's a database you can all access already, where you can see what the world is working on already in terms of microbial proteins. So that's the value chain tool.proteinclub.eu. But within this project, we will also make a database of microorganisms that are very suited to produce proteins, how you can modify them to produce even better and more proteins. In another work package, we will make techno-economic assessments for the production of biomass protein from side streams. And we're organizing events, such as the one on Thursday at the University of Ghent, and it's called What's Shaking in Protein Research. You can still register. It's even for free, I think. And it's not so far from here. So what are the specific challenges when you produce or scale up proteins? You can see a picture at the top of ovalbumin. It's a very complex molecule. So, but it's actually the complexity or the conformation of these molecules that determine these very valuable applications and properties. But they are difficult to analyze, they're quaternary structures, and they will determine your foam stability or the stretchiness of your cheese. These properties are impacted by the medium composition of your process and by the parameters you're processing them at. You can ruin them by irreversibly denaturing them when you process them too harshly or you don't treat them nice. You can have some protease activity wandering around the process that basically breaks down your protein and you don't find it back at the end. You can have issues with microbial stability because proteins are very tasty also for other microorganisms. Those great functional properties that you can see on these slides, like the foaminess, the stretchiness, the viscosity, the emulsification properties, gelation properties, can cause a lot of issues during processing, as you can imagine. You can have Maillard reaction between amino acids and sugars and have off flavors or decoloration of your product. Off flavors can also occur from enzymatic degradation. Proteins can be allergenic, so you have to take care of safety during processing. And the complexity and diversity of all these applications make scale-up difficult to predict. You cannot simply model it and be sure that it will behave in the way you had predicted it. You really have to test this to strengthen your models. So how do we tackle these issues while we scale up? First of all, you need state-of-the-art equipment. It has to be designed to allow sanitation and good maintenance, sound procedures. You have to have this equipment at a very small scale to de-risk in a cheap stage. And you have to demonstrate at a relevant scale. You need all the analytics in place to monitor the process online continuously and 24/7. You need the technological know-how. It's not enough to have a unit. You need to know how to work with it and how to work with it efficiently and to tweak all the parameters. And that's what our engineers and technicians do day in, day out. You need a very structured preparation of your trials. You need very clear objectives. Do I want to optimize or do I want to produce a certain amount of product? You have to assess the risks of your process. You need a sampling and analytical plan upfront, so you learn as much as possible from the trials you're doing. And you need backup plans, because you can be sure that the products will behave differently than you had anticipated. When you start doing fermentation, of course, you need an affordable feedstock. First-generation sugars will work great, but maybe side streams are a better economic choice. You need a robust seed train that is also very reproducible. You have to choose, am I going to run a fed batch or a continuous process? And what is my feed strategy? You need an aeration strategy, foam control, and sterility. In purification, you have to make sure your process is as simple as possible and as complex as needed. If you have unexpected events, you have to make sure to do a root cause analysis to learn from everything that goes wrong. and you have to generate data. This entire data package that we generate throughout the processing scale-up is also delivered to our customers. Because scale-up and bringing an innovative process to the market is always a joint effort, we also organize an event that's called Pitch Perfect. We had last year 100 companies, startups, pitching their innovation to over 50 investors that reverse pitched their programs to those startups and matched them there. We had 450 attendees, so please be welcome, 25th of November in Brussels. And again, to show that it's all teamwork, this is our team in daily contact with all of our customers. And that's where I thank you for your attention.